Chinese community, and then all of you. It's fantastic to see such a large group of people here. Also your new GSR. Yeah. Sitting under there. Yeah.
I do.
It's absolutely brilliant seeing all of you turn up for Charlie's Defence. It's always for me a sad day as well as a happy day, because I know at some point it means goodbye, right? And it's been such a pleasure working with you, really. Okay, so let me talk about Charlie's achievements. Okay, we've got a long list, so I actually had to write them down. So during Charlie's PhD, he's written 5 papers. They're published. He's got another one on the way, and actually very likely another one that's coming through by the end of summer. His first paper that he published actually won the AIRFRB. So this is actually research board for the best student paper of the year in 2015. Right? He's given talks in over 15 conferences. His collaborations have actually expanded quite dramatically. So when Charlie first arrived, we basically told him, we've got this really big project, and it means that you have to get on with everyone, and you have to talk to everybody, and you have to go and work at this hatchery. And the collaborations included the state, the tribe, and federal scientists. And Charlie was actually really good at liaising with everyone and getting his research project going, and getting all the samples that you need, and going and meeting with them and giving presentations. And so from this original collaboration, essentially he started then working with international researchers. So he spent time in Finland as well on another paper, which we hope will be done by the end of summer. We believe will be done by the end of summer. And then very recently working with Tim Thornton on a human data set. Right? So Charlie's experience is really extensive. Awards, I had to write these down, right? Because there are so many. And I've actually separated them by the level that you got your awards at. So at the college level, you got the Hall Conservation Genetics Award. You got 3 scholarships in SARS, and you got an international travel grant. At the national level, Charlie won a C grant Population Dynamics Fellowship to work on the relationship between inbreeding and population viability with Jeff Hart, who's on his committee. The Marine Technology Society Graduate Student Scholarship. And then an honourable mention from the Skinner Award at AFS level. At the international level, right, so we don't start just nationally, he got the Joint Fishery Society of the British Isles AFS International Section Fellow Award. And as a result of that, has actually worked on international collaborations and international exchange with a group of students. And then most closely and dear to my heart, Charlie's just been awarded the Faculty Merit Award at the PhD level, so very proud of this, Charlie. You've been a really active member on a whole bunch of things within the school. So Charlie's basically served on the various student committees. He was on the faculty search, where essentially he was asked to control all the IT stuff because the faculty were completely hopeless at it. You've worked with Sea Grant on the Orca Bowl, whole bunch of outreach, which is too numerous to mention. And then also worked and volunteered with the UW chapter of Engineers Without Borders, where he basically volunteered to go work in Guatemala on helping people establish agricultural facilities. Okay, so on a personal note, Charlie, it's been such a pleasure having you in the group. We've got a wonderful group, very proud to have this group of collaborators, students, my colleague Lawrence Hauser, and you've been key to this group. We've got a wonderful culture, and it's made us proud, like being part of this interaction between us. You've been really good humored from the day that you arrived, when you were the junior scientist and everyone was attacking your computer. To allow your picture to be submitted every year for the annual SAS t-shirt. To be the first person to suggest the faculty dunk tank at the podium.
Oh, I think that's Dave.
Oh, sorry. Being involved in the dunk tank. Yeah. On a personal note, I've learned so much from you, and it's been such a pleasure working with you, and it's really good to be here today. So it's all yours.
you so much for that introduction. It's a pleasure to be here. Thank you all for coming and taking time out of your busy schedules. Yeah, today I'll be talking about my dissertation research, which has been going on just for a few years now. But yeah, evaluating the effectiveness of managed gene flow to reduce genetic and phenotypic change associated with captive breeding in Chinook salmon. I just want to acknowledge that this project, as Kerry alluded to, was a very collaborative effort between multiple organizations. See if I can use this right. Yeah, so I just want to acknowledge them right off the bat, but I'll of course have more acknowledgements at the end of the talk. So let's see. Start the timer so I've kept my presentation to about 2 hours. All right, so captive breeding is a commonly used tool for conservation for both declining populations and also threatened and endangered species. And it can be an effective tool because they bring individuals into a very benign captive environment. And so the goal is to reduce or increase the survival of those individuals compared to their survival rates out in the wild. And then ultimately try to rebuild that population and hopefully reintroduce individuals into the wild to rebuild the populations. The salmon hatcheries are it's a form of captive breeding that's used to supplement declining wild populations. And in these programs, adult salmon are brought into the hatchery. They're artificially spawned, and their juveniles are raised for some period of time, typically a year or two. So that depends on the species. Before those juveniles are released into the wild. And so for conservation purposes, the goals of salmon hatcheries are to take a small population and both increase the size of that population and also provide surplus fish for harvest. And so therefore you're redirecting the fishing pressure from the wild population to the hatchery reared fish. Now, salmon hatcheries, there's some evidence that shows that hatcheries have been beneficial and provide a short-term demographic boost to the population. That being said, the magnitude of that boost and the duration of the boost needs more research because the magnitude can be minimal in some situations and the demographic boost can end sometimes after the hatchery program stops. So the long-term effectiveness of hatcheries to support the wild populations is still a very active area of research. In addition, there's a lot of ecological and genetic risks associated with hatcheries. Today I'll just focus on the genetic risks. The first being domestication selection. Now domestication can occur because captive conditions are very different from the wild environment. Captive conditions are very benign and they're very stable. So this can cause a relaxation of natural selective pressures. So there's no mortality or no predation in the hatchery and abundance of food. So this can allow individuals to survive in the hatchery where they may not otherwise survive in the wild. And in addition, there's selection that may be imposed, whether it's deliberate or inadvertent, selection that's imposed that could favor individuals that are suited for captivity, but they may not be suited for the wild. So overall, domestication may decrease the fitness of hatchery-reared fish when they're released into the wild. The next risk is genetic drift. And you can think of genetic drift as sampling effect. So it's random genetic change over time. And to borrow one of Lawrence's examples, let's say you have a bunch of smiley faces, different colors, and over time, some smiley faces reproduce, some do not. So over time, you get, based on this random genetic change, you can get a loss of genetic variation within a population, and also divergence between populations. And the loss of genetic variation here is denoted by the increase in blue smiley faces. And importantly, this forest genetic drift is stronger in small populations. So here are two plots showing the frequency of an allele at a locus on the y-axis and time in generations on the x-axis. And each line here in these plots represents a population that comprises either 10 individuals or 1,000 individuals. So as you can see, there's a lot more random fluctuations of these alleles over time in these different populations, in the smaller populations as opposed to the larger populations. And this is important because, again, it can decrease genetic diversity within a population, and that in turn can decrease the potential of the population to adapt to future change. So the last genetic risk is inbreeding. So I'm from North Carolina. So I know, but in case you don't, I've used that joke at least 3 times. So if mating between related individuals and it, like genetic drift, is more likely to occur in small populations. And it can also have negative effects on the fitness of these organisms, and this is known as inbreeding depression. So as a quick example, here's a survival of Drosophila on the y-axis and increasing levels of inbreeding on the x-axis. And as you can see, the survival decreases with increasing levels of inbreeding. So this is an example of a negative effect of inbreeding on fitness. So together, these genetic risks can decrease the fitness of hatchery fish when they're released into the wild. And specifically, this has been most commonly documented as hatchery-reared fish do not produce as many offspring in the wild as fish that were born in the wild. And this has been documented across multiple species and multiple systems. And so this issue is very important when you consider that over 2 billion hatchery salmon are released every year across Western North America. Now, not all of these are released for conservation purposes, but nevertheless, it's a very, very big issue. So one of the potential solutions that's been adopted actually to try to mitigate these risks is the use of integrated management, or what we call managed gene flow. So salmon hatcheries can be operated traditionally under 2 dominant regimes. The first is more the traditional practice called segregated hatcheries, and then this new approach, integrated hatcheries, that's been adopted over the past 15 years or so. So in both of these programs, adult fish are brought into the hatchery, and they're artificially spawned, and their juveniles are raised for, offspring are raised for a year or 2. Then they're released out to the ocean. But with segregated hatcheries, when those hatchery fish mature as adults, they're brought into the hatchery to produce the next generation in segregated hatcheries. So for these, all the brood stock comes from hatchery origin individuals. Now with integrated hatcheries, the hatchery-reared fish are allowed to spawn in the wild, and then the wild fish are brought into the hatchery, or at least a proportion of them, and they're used as brood stock to produce the next generation. So in theory, segregated hatcheries have no real gene flow with the wild population, at the very least from the wild into the hatchery, whereas integrated hatcheries have 2-way gene flow. And in theory, as long as the proportion of wild fish that are brought into the hatcheries exceed the proportion of the hatchery fish that are spawning in the wild, then the population as a whole will be driven by select or evolutionary processes that are occurring in the wild, in theory. And so integrated management has been adopted over the past 15, 20 years, and it's been based on a lot of theoretical studies. We have our own Mike Ford in the audience. He was one of the initial studies. So these theoretical studies suggest that managed gene flow or integrated management can reduce these risks. However, it's never been empirically tested in a comparative setting. And so that's what my dissertation aimed to do, to test the effectiveness of managed gene flow when compared to the traditional segregated management to reduce genetic and phenotypic change that may be associated with captive breeding. So we had 4 chapters for the dissertation. First asks overall, does managed gene flow reduce overall genetic divergence when compared to the segregated one? Secondly, does managed gene flow reduce the genetic and phenotypic effects of inbreeding specifically? Third, does managed gene flow reduce divergence at loci that are associated with traits that are related to fitness? Because these traits are ultimately the traits that matter for the fish in the wild. And then the 4th chapter, which was a sort of last-minute switch, but it stems from the 3rd chapter, is by identifying trait-associated loci. We learned a lot of things throughout that process, and so we provided some recommendations, or at least some considerations for other people who are looking to identify important loci that are associated with traits. I won't talk about the 4th chapter today, but if you want to read about it, let's say on Friday night, you can download the paper and just have at it. But for the other 3 chapters, we're using spring-run Chinook salmon at the Cleveland Supplementation Research Facility to address these questions. And this facility is really unique because it maintains both an integrated and a segregated hatchery line. So in the late 1990s, for us, 1998, the wild population returning to the system was essentially divided into 2 components. So half the fish, approximately, were brought into the hatchery and spawned, and their offspring, so the offspring of those wild fish that were brought into the hatchery, for our purposes, they're the 1st-generation hatchery adults of the segregated line. And then the other half of the wild population in 1998, those fish were allowed to spawn in the wild, and then those offspring were brought into the hatchery in 2002 That's sort of, for our purposes, the initiation of the integrated hatchery line. And then over the years, the segregated line continually spawns just hatchery origin fish year after year. And so there's, again, no gene flow with the wild population. Whereas the integrated line, the hatchery reared fish are allowed to spawn in the wild, and then wild or natural origin fish are brought into the hatchery. So this is a good time to say that natural origin fish are fish that were born in the wild, but they may have some proportion of hatchery ancestry, because hatchery fish from this line are allowed to spawn in the wild. And so we have a really nice experimental design to compare the integrated and segregated lines over multiple generations, compare them both to each other and to the surrounding wild population. So just to provide a little more background on the system, so the Cle-Elum Supplementation and Research Facility is operated by both the Yakima Nation and the Washington Department of Fish and Wildlife. And the Yakima River, the spring run Chinook salmon there, it's a tributary of the Columbia River, and all returning adults have to pass through Rosa Dam. And here they have tissue samples taken for DNA, and they also have a bunch of measurements taken, like length and weight. And then, I guess I should note that the integrated and segregated hatchery lines, fish from those lines are differentially marked. So they're able to tell which fish is from the segregated line and which fish is from the integrated line. So only hatchery fish that are from the integrated line and wild fish, they're passed over the dam and allowed to continue their migration to the spawning grounds. But then both integrated line fish and segregated line fish that are used as brood stock are collected at Rosa Dam and they're trucked to the Cle-Elum facility. And there they're artificially spawned using 3x3 factorial matings. There's a picture of the hatchery. And then the juveniles are raised at the hatchery for about 16 months. And then the juveniles are transferred to 3 acclimation sites. And these acclimation sites were designed to increase primarily the spatial influence of supplementation efforts, because this program was largely created to supplement the declining population in the Yakima River. And then after about a couple months at the rearing facility, at the acclimation sites, the fish are allowed to begin their migration to the ocean. So I mentioned a bunch of data that's collected at Rosa Dam, and I just want to describe some of that data. So we have phenotypic data. So both at Rosa Dam and at Cle-Elum, there's a bunch of different traits that are measured. So both at Rosa Dam and Cle-Elum, we have length and weight of the fish. And we can also calculate condition factor, which is a composite measure of length and weight. And that sort of gives us an idea of how fat the fish is. We also have the day at which the fish return to Rosa Dam. We consider that to be return timing. We also have the age of the fish when they return to the system as mature adults. Then at Cle-Elum, we also have length and weight. We have maturation timing, or the day at which they're spawned in the hatchery. And then based on some work by Kurt Knudsen and others, we also looked at daily growth coefficient, which you can think of essentially between the time when the fish is measured at Rosa Dam and when the time the fish is spawned at Cle-Elum, which is the rate of weight loss that the fish experiences while they're being held in the hatchery. And then for females, we also have fecundity, so the number of eggs they have, and then also reproductive effort, which is the proportion of the body mass that's devoted to gamete production. So those are our phenotypic data that we have for our chapters, my chapters. We also have genetic data, so tissue samples were collected, and so we sequenced the DNA of those tissues using a process called restriction site-associated DNA sequencing, also known as RADseq. We took those sequences and we aligned them to a baseline about 50,000 loci that was developed by Marine Review for Chinook salmon. And this enables us to reliably identify loci for a number of reasons, which I won't get into here. And importantly, about 7,000 of these loci are mapped to the genome. So this is important because if we observe a signal of some genetic signal, let's say a domestication selection, if all those loci are mapping to the same genomic position, that gives us more confidence that what we're observing is a true signal rather than potentially a false positive. So we aligned our data to this baseline, and we filtered it for a variety of parameters. And for the most part, we're using about 9,000 polymorphic loci. And by polymorphic loci, we simply mean a place in the genome where there's a nucleotide difference between individuals. So that's largely the data, both phenotypic data and genetic data, that I'll be using for the rest of my dissertation. So we go to the first study. Does managed gene flow reduce overall genetic divergence that may be associated with captive breeding. So to achieve this, we specifically compared the integrated and segregated hatchery lines across 4 generations. We compared them to the wild founders using those 9,000 genetic markers. We identified differences between the lines, including signatures of domestication selection. And the ultimate goal is to determine if integration was an effective practice at reducing divergence that may be associated with captive breeding. So the results of this study, maybe for a Saturday night, if you want to, they were published a few years ago. All right, so with this chapter, just to remind you, so we have our sampling designs. We actually sampled approximately 50 to 60 individuals for the wild founders, and then each generation of the integrated and segregated hatchery lines, with the exception of the F4 generation. Due to financial constraints, we could only sample approximately 25, 28 individuals. And I guess these are the individuals that were left in our final data set. So we sampled a lot more and tried to sequence a lot more, but these were left after we filtered for missing data and things like that. So we're comparing each generation of the 2 lines to the P1 founders, the wild founders, using these 9,000 markers. So we first conducted a discriminant analysis of principal components. And this is a multivariate technique that's used to summarize genetic variation in 1, 2 dominant axes of variation. And it allows us to investigate genome-wide divergence between these different groups. And then we investigated mechanisms that potentially were driving any divergence that we observed. So the first thing that we did was we estimated effective numbers of breeders. This gives a proxy measure for genetic drift. And then lastly, I identified signatures of domestication selection using 3 independent methods. So to dive into the results of the discriminant analysis of principal components, we find that gene flow does indeed produce genetic divergence. So here we have a density plot of individuals along the dominant axis of genetic variation for the DAPC. And this is our P1 found group, so the wild founders are in gray. And when we overlay the different generations of the integrated and segregated hatchery lines, they're denoted by shades of blue and red, respectively. We see that in the first hatchery generation, both hatchery lines cluster closely with the P1 founders. But then as we progress to the second, third, and fourth generations, we see that the integrated line, each generation, clusters closely together with the P1 founders, whereas the segregated line slowly diverges over time. And if we calculated the level of genetic divergence, so this is FST, where a higher value indicates a larger degree of genetic divergence from the wild founders. We see that divergence in the segregated line was approximately 4 times the level that we observed in the 4th generation of the integrated line. So this is really interesting because it provides, I guess, an insight into the rate of divergence that's possible with these 2 management approaches. And as a point of comparison, this level of divergence in the segregated line, if it's propagated for another 3 to 5 generations, the segregated line will become more divergent from its founding population than we would observe between 2 wild populations of Chinook salmon. FST and noted between 2 wild populations of Chinook salmon, at least for some, ranges from 0.02 to 0.05. So it really provides insight into the rate of divergence. That's beneficial for evaluating the risks and benefits of hatchery programs over time. So next we wanted to investigate what are the mechanisms, what are the processes that are driving this divergence. So first looked at genetic drift. And as I mentioned earlier, it's more of a random genetic change over time, and it's stronger in small populations. And I estimated effective number of breeders, NB, as a proxy measure of genetic drift. So you can think of NB for the purpose of this talk, you can think of NB as essentially the genetic population size of a cohort of salmon. So let's say we have 2 populations of salmon and each have 1,000 individuals total. Let's say all 1,000 individuals in population A contribute genetic material equally to the next generation, whereas only a fraction of individuals in population B contribute to their genetic material to the next generation. So even though they're the same total population size, their NBs would be quite different. And so we would expect that genetic drift would be larger in a smaller genetic population than in a larger genetic population. And this, we investigated this because the integrated line spawns more fish than the segregated line, about 4 times as many fish. And this is due to the objectives of the CLELM program. Their main objective is to supplement the wild population. So given these differences in broodstock, we expected genetic drift to be lower in the integrated line, and it would be larger in the segregated line. So we estimated genetic drift using the linkage disequilibrium method for eugenesis. And what we observed, we did indeed observe that the integrated line had larger levels of NB than the segregated line. And even after correcting for those differences in broodstock sizes, we still found that the integrated line increased NB relative to the segregated line. And this is pretty much an artifact because the integrated line uses both the hatchery environment and the wild environment because they're a single connected population. They have both of those rearing spaces, and so it's not a surprise that we observed a larger NB. And this suggests to us, given these differences, that genetic drift is driving divergence in the segregated line. As some of you may have observed, the NBs in the integrated line started to decline from the F2 to F3 and F4 generation. Well, we have to standardize based on total population size in those generations. And so when we standardize those estimates of NB to the census size, the number of fish that are in the system, we actually see that the ratio of NB to total population size actually increases from the 2nd to 3rd generation, but then it also decreases from the 3rd to 4th generation. So to me, this really stresses the importance of monitoring both ecologically, total number of fish entering the system, but also genetic monitoring over time, because you may not otherwise see these various trends or deviations if you do not conduct both. Then the last thing I want to note about this slide is the NBs of these two populations, essentially the first integrated and first segregated population. Now these populations, these NB estimates, were derived from the same number of fish. So approximately half the population was allowed to spawn in the wild, and this represents the NB of those fish. And half the population was brought into the hatchery to initiate the hatchery, and this is the NB of those fish. So even though approximately equal numbers of fish were allowed to spawn in the wild and brought into the hatchery, we observe a decrease in NB in that first generation hatchery fish. So this may suggest that simply bringing fish into the hatchery may have caused increased variance in reproductive success or factors that decrease the genetic population size, which is of course a risk of hatcheries. So we think that genetic drift may be driving divergence, but we also investigated signatures of domestication selection. And whereas genetic drift is random fluctuations over time, we consider domestication selection to be, we would expect it to be more consistent over time because the hatchery environment is imposing very stable conditions generation after generation. And so we looked for signatures of domestication selection by comparing each generation of the hatchery lines to the wild founders, and we did this using 3 methods. The first was a moving average of divergence, and that was used to identify regions of the genome that may be significantly elevated, experiencing domestication. And we also used 2 methods, F-temp and base scan, that were used to identify specific genetic markers that may be experiencing domestication selection. So as an example of how this would look, here we have FST, which again is a measure of genetic differentiation. And this is FST of each generation of the hatchery line to those wild founders. So we have the segregated line on top and the 3rd generation of the integrated line on the bottom. So the red line represents the moving average of divergence, and the gray shaded region represents the 95% confidence intervals of our null expectations. And so any region where the red line exceeds the gray shaded region would be considered a region of elevated divergence, and that may be indicative of domestication selection. And then with these 2 other methods, F-temp and base scan, we could identify specific genetic markers that may be experiencing domestication selection. And we placed emphasis, when we did these analyses, we placed emphasis on areas where all 3 tests overlapped. So we have a region of elevated divergence here, and we also had a locus within that region that was identified by these other methods. So that provides more support for a true signal. We hypothesized that we would observe more outliers, more of these regions, in the segregated hatchery line because this line is continually exposed to the hatchery environment generation after generation. And lo and behold, we did observe that. We observed more consistent evidence of divergence in the segregated hatchery line. So here again, we have FST compared to the wild founders along the y-axis, and x-axis we have position along a chromosome, and this is chromosome 12. We have our first generation of the hatchery line, so this is first generation of the segregated population, and this is first generation of the integrated population. And if you pay attention to this region that I've outlined in the black box, we see that over time, there's a consistent elevated divergence in the segregated line that increases over time, with the exception of the fourth generation. Whereas in the integrated line, this region is significantly elevated from the second generation, but then it goes back down. So it's not as temporally consistent. And so in this region in particular was supported by all three methods that we used in the segregated line, and it was not supported by all three methods in the integrated line. So this is an example of a region that may be experiencing domestication selection because we're observing temporally consistent divergence in the segregated line, but not in the integrated line. So as a summary, we observed for the moving average test, for regions that were consistently divergent in 3 to 4 of the generations, we observed 7 regions in the segregated line, but 0 in the integrated line. For our locus-specific tests, we observed more outliers in the segregated line for this other F-temp method. And then for base gain, we observed equal numbers of loci, but that was simply because to achieve a power to run this program, we needed to combine all the populations together. So that's why these are equal. But overall, these represent, particularly 3 years ago when these results were published, they represent some of the first signatures of domestication selection identified in Pacific salmon. So these are certainly starting grounds for future investigations. And in these regions where, for example, in the previous slide in chromosome 12, where there was overlap between the various tests, we annotated some of the genes in those regions. And we found a few genes that were related to the processing of proteins, lipids, and sugars. This is very broad, but those genes have been shown in the literature. Their expression has been shown to be altered by the food availability and composition in the hatchery environment for rainbow trout. And similarly, 3 genes related to protein degradation, but also their expression was also altered by food. Then we had 2 genes that were related to the clustering of these receptors called GABA receptors. That's actually very intriguing because GABA receptors are involved in gonad growth and early maturation. And one of the common issues with hatcheries is a higher incidence of early male maturation in the hatchery. And so these are certainly starting points for future investigations that could provide insight on that phenomenon. So there's possible links to differences in food availability, composition, and maturation differences between the hatchery and wild environments. All right, so chapter 1 in summary, we did indeed demonstrate the effectiveness of integrated management to reduce genetic divergence. We identified genetic drift as the prominent or dominant mechanism driving the divergence, as noted by the low MDs of the segregated hatchery line. But we also identified regions that may be experiencing domestication selection in the hatchery. And these were more consistent in the segregated line. And there's possible links to food and maturation when we annotate, of course, require more exploration. All right. Chapter 1. Obviously. The literature, the theory suggests that managed gene flow was effective, but we were the first to empirically demonstrate it. So I think that's really exciting. So now on to inbreeding. Chapter 1, we investigated genetic drift and domestication, but now we're going to look at the other genetic risk of inbreeding. So specifically, we're going to estimate inbreeding coefficients in the founding wild population and each generation of the integrated lines. We're going to compare levels of inbreeding over time. We're going to quantify the effect of inbreeding on 8 traits. And then another thing that I wanted to mention, but we're not going to talk about here, is that an integral projection model to determine the effects of fitness and how that may affect the supplemented wild populations. This is the work that I'm Jeff, and we got an uphill battle, but we're going to do it. So I'll just focus on objectives 1 and 2 right now. Just to remind everyone, Inbreeding more likely to occur in small populations, and so the segregated line is particularly a small population, so we would expect potentially higher levels of inbreeding in that population. And inbreeding may negatively affect fitness. And we measure inbreeding, or we estimate it by this measure called F, and that estimates the portion of the genome that is identical by descent. So just to remind everyone of our sampling design, we have 50 to 60 samples, few generations, then 25 samples for this F4 generation. And due to various filtering parameters, when I conducted this analysis, we're actually only using 6,800 loci this time instead of 9,000. When we're integrating this genetic data, we're going to tie it into the phenotypic data. So we have length and weight measured at Rosa Dam, condition factor of the fish when it returns to the system, so sort of how fat the fish is, the day of return to the system, return timing, and then we have spawn timing, daily growth coefficient, which again is sort of the rate of weight loss between when the fish arrives at Rosa Dam and when the fish spawns at the hatchery, and fecundity and reproductive effort for females. So we estimated levels of inbreeding in each hatchery line, and then we estimated the effect of F on fitness, on those fitness-related traits. So here we have levels of inbreeding on the y-axis, and for these measures that I implemented, it can be negative. That just simply means the individual may be outbred rather than inbred. So we have the P1 founders here in gray, and then each generation of the integrated line and segregated lines on the x-axis. And the first thing that strikes me is really the levels within a generation, so within the F1 generation, the levels between the two hatchery lines are not that different from each other. And in fact, they're only significantly different in the F2 generation. So only this generation, the segregated line, has a significantly higher level of inbreeding than the segregated line. And to me, when you consider the NB estimate of the previous chapter, so even though these populations have very different NB estimates, station sizes, the levels of inbreeding are relatively the same. So actually, for management purposes, reassuring things. So maybe inbreeding may not be a genetic risk that is as important as other genetic risks, but there's some caveats. I want to point out is in this F1 generation, you can see that the levels of inbreeding in both the F1 wild and the F1 hatchery fish, they're both significantly higher than the levels of inbreeding in the P1 founders. So my best guess, this increase in both of these lines relative to the P1 founders was that wild population was split into two at the initiation of the hatchery. So half the fish were brought into the hatchery, then half the fish were allowed to spawn in wild. So you're taking that wild population, which had already experienced a few years of low numbers of fish returning to the system, and essentially cut it in half, increased the risk of inbreeding in these two populations. Then I'd also like the levels of inbreeding in the F4 generation. So these levels of inbreeding are significantly lower than the previous levels in both hatchery lines. And this could be due to a variety of reasons. So one, these populations have about half the number of samples than the previous generations, so it could be a sampling effect. We also explored any issues with our data in this F4 generation with Gary and Creed's help and Carolyn's help, but we did not actually see any differences in our data in this F4 generation compared to the other generations. But then also the other possibility would be maybe inbred fish, when they were in the ocean, maybe they experienced a higher level of mortality in the ocean in this particular year. And so these fish returned in 2014. That corresponds to the initiation or the observance of the warm Pacific, the blob, that warm water mass that had negative ecological effects. So one of the theories that I came up with one of the hypotheses was maybe that those harsh ecological conditions in the ocean, maybe that caused a lower survival of inbred fish compared to the non-inbred fish. But there's no way to tell right now, at least. So then, given these levels of inbreeding, we investigated the effect of inbreeding on these various fitness-related traits. And so for each of these traits, we modeled the trait value as a function of inbreeding coefficient We incorporated a quadratic term, inbreeding squared, to try to account for any nonlinear effects. We've also various sort of interactions. So for each of these various code model selection to identify the best model. And from there, we estimated, we quantified the effect of inbreeding on these various traits. So I'll try to walk you through it. So for each of these 8 traits, the check mark denotes the populations or the group of fish for which inbreeding significantly affected trait value. So for example, inbreeding significantly affected spawn timing in the integrated hatchery line, both males and females, across all 4 generations. And as a counterexample, condition factor was affected by inbreeding in both hatchery lines, both males and females, but only in the F1 generation. These different generations, the fish experience different environmental conditions, and so we may or may not expect the effects of inbreeding to be the same across all the generations. So we see that inbreeding affected 4 of the traits, some only in the integrated line, some only in males rather than both sexes, and there was definitely a temporal variation. So the fitness effects of inbreeding varied by hatchery line, sex, and generation. And to give an example of what this looks like, here we have spawn timing, so the day of maturation of the fish on the y-axis. We have inbreeding, so lower levels of inbreeding to increasing levels of inbreeding on the x-axis. Then we have our empirical data points in black, and then we have our model projections. So model predictions for each generation represented in the solid lines with 95% confidence intervals. And actually here, inbreeding caused a significant positive effect on spawn timing. So inbred fish in the integrated line in males, I should note that, those fish matured later than the non-inbred fish. Now, whether or not this has a biologically positive or negative effect, we do not know at this point. But it was also observed that the positive trend was observed for females, but it was not observed for the segregated line, males or females. Anyone has any theories as to why we observed it in the integrated line as opposed to the segregated line? I'd love to hear them. So I have no idea. 2, in summary, inbreeding remained relatively low over time for both the integrated and segregated hatchery lines. And this was surprising to me because the 2 lines had large differences in the genetic population sizes. But there's some evidence that managed gene flow reduced these risks. We looked at pairwise relatedness too, that influences future levels of inbreeding. And also in that F2 generation, the segregated line had significantly higher levels of inbreeding. So there's some evidence that inbreeding, those risks are mitigated by managed gene flow. The effects of inbreeding on fitness varied and I think requires more exploration. Particularly, we sampled adult fish, so returning adults. So those represent the survivors. So the small fraction of fish that survived all like the freshwater, the marine environments. And so those fish are really, you could think of it as the most fit fish. So we didn't observe any differences between the lines, or minimum differences in the adults, but let's sample the juveniles to see if there's differences between the hatchery lines and to see if we could then address the question, are inbred fish experiencing higher levels of mortality in the marine environment? So does managed gene flow reduce genetic and phenotypic risks of inbreeding? Possibly. We need to revisit that later. Then for the last chapter that I'm going to present, does managed gene flow reduce divergence at trait-associated loci? And these are the loci that are important for fitness. So specifically, we're going to first we need to identify loci that are associated with 6 traits. So we actually did this study before the inbreeding study, before I knew that fecundity and reproductive effort were traits for which we had data. So we only identified loci associated with 6 traits. Then we compared the integrated and segregated lines at those loci, and then we also compared trait-associated loci to our previously identified outlier loci that I alluded to in chapter 1. And ultimately, we determined if managed gene flow limited divergence, And the results of this study are also published. They were published earlier this year, so you can look at that paper for a more in-depth explanation and more detailed results. So I conducted this before chapter 2, so we're still using about 9,000 loci now. And we're exploring traits, loci that are associated with 6 traits. So we have weight and length, return timing, and age of maturity. Then we have spawn timing and daily growth coefficient. So we do not have in this chapter fecundity and reproductive effort. So I just want to introduce the basic premise of how we identify loci that are associated with these traits. So let's say we have a chromosome here, and we have different markers represented by black lines. Let's say we have one genetic marker, or if a fish has a nucleotide, it's really big. And if the fish has a T nucleotide, it's really small. That's, of course, like the most simplistic example that I could give, but it's not going to be that simplistic, but this is an example. So we're looking for loci that may be influencing the size of the fish or the trait value. So to identify these loci, we actually used an algorithm called random forest. That's a really powerful algorithm because it takes your phenotypic data and your genetic data, and it builds a series of trees that partitions the phenotype based on the genotypes and the different loci. So it identifies, let's say we have 500 fish that are resistant to a disease and 500 fish that died when exposed to a disease. And so random forest will identify the SNP that best partitions those two groups of fish. And so this partition, if the fish had this genotype, there was more susceptible fish than resistant fish. Whereas if the fish had this genotype, more of them were resistant and less of them were susceptible. And so random forest builds a series of trees, continually finding loci that best partition the response variable or a phenotypic trait. And so this is a very powerful approach to identify groups of genetic markers that together explain variation and can partition variation of a phenotypic trait. So it can incorporate interaction, whereas other methods may not be able to incorporate interactions between loci. And this figure is from Marine Brew and others, my paper, and Kerry and Dan. Anyway, moving on. So we identified loci with these 6 fitness-related traits. We identified a number of loci associated with each trait, and the numbers of these loci that were mapped to the genome are in parentheses here. And then this is the percent variation explained by these groups of loci. So you can see that for all of these, so these 5 traits are continuous traits. And so this is analogous to, you can think of it as the R squared, so how much variation in the trait did these loci explain. And these are all relatively similar. And we think that that's because we're using essentially the same data set for each of these traits. And so we're not surprised that we're observing the same amount of variation explained. But you'll also note that there's a lot of variation that's not explained in our data. So that's a missing variation that we have yet to identify. Then age of maturity is a categorical variable. Fish can be 3 years old, 4 years old, 5 years old. And so this is the OOB error rate, represents the out-of-bag error rate. It's simply a measure of how well can a fish, like what is the error rate for a fish being classified if an age 4 fish is run down the random forest tree, how often is it misclassified as an age 5 fish or an age 3 fish? So it's misclassified about 25% of the time. Anyway, so if we look at the predictor loci here, so these groups of loci, specifically the loci that are mapped to the genome, if we plot those along the genome, so here we have cumulative linkage map position in center orbits. So you can consider this as position along the genome, and each of them are denoted by alternating shades of white and gray. So this is chromosome 1, chromosome 2, all the way to 34. We have the loci denoted on top. Diagram of the there are, I think it's 5 centimorgan bin as you move along the chromosome or along the genome. And so what we can see here is that the trait-associated loci are scattered throughout the genome. But if we take a closer look, we do identify regions of the genome excuse me, that may be candidates for regions that are involved in fitness. So regions where trait-associated loci cluster together. So here we have two chromosomes, chromosome 8 and chromosome 10. So as an example, we have two loci, one associated with return time and one associated with spawn time, that are both mapping to approximately the same position. These two traits can be correlated in some populations. Then we have two loci associated with spawn timing here in the same location. So this provides more evidence that this region may be important for spawn timing. And similarly, like fork length and weight are very highly correlated in this system, as probably in most systems. We have multiple loci that are associated with these traits, all mapping to the same genomic position. And then these regions are further supported when we incorporate loci that were identified in other studies of Chinook salmon. So for these purposes, I'll just present two. So we have Kathleen O'Malley and others identified a locus that was associated with broad seasonal runtime of Chinook salmon, so fall or spring run Chinook. And this locus, actually, if you follow the line, it clusters closely with spawn timing. And so broad scale run timing and spawn timing can be correlated. So that provides more evidence that this region may be important for that trait. Then also, Marine Review identified a number of loci that were associated with runtime as identified by peak run month of the fish. And so all of these loci on the end of chromosome 10, they cluster closely together with fork length and weight. And these traits can be correlated in some systems. And then similarly, one of her predictor loci for run timing clustered closely with our loci, one for spawn timing and one for run timing. So when we look at the mapping, the positions of these loci, and we incorporate loci from other studies, we can identify regions that are potentially involved in fitness that can be candidates for future sequencing efforts. Then we want to link traits with domestication selection. We want to explore, are any of these traits potentially experiencing domestication selection? So we did this in a number of ways. We first quantified phenotypic differences between these traits, between the integrated and segregated lines. These could be due to environmental factors or genetic factors. But we quantified traits because if a trait was phenotypically different between the two hatchery lines, that would certainly provide more evidence that, hey, this trait may be experiencing genetic change in the hatchery. So for each trait, we have the difference between the segregated and integrated line females and segregated to integrated line males. And the few traits that I want to point out: return timing. Segregated line males returned significantly later to the system than integrated line males, approximately 11 days. And in addition, for weight, segregated line, both males and females, weighed less than integrated line fish. And interestingly, for spawn timing, both segregated line males and females, they matured earlier than integrated fish. So the males returned later, but matured earlier. So there's really a compression of the spawning season that we observed in these hatchery fish. And the standard deviations for return timing of the population as a whole are about 20 days, 19 days for return timing, and the standard deviation of the population for spawn timing is 8 days. And so this is approximately half the standard deviation difference between the hatchery lines, and this spawn timing is over a full standard deviation difference. So these could have very significant biological implications. So we observed differences in a number of traits between the two hatchery lines. Then we compared the two hatchery lines using a multivariate approach called principal components analysis, using only the loci that were associated with the traits. Because similar to the discriminant analysis of principal components in chapter 1, if we see genetic divergence at trait-associated loci, that may be indicative that the hatchery lines are diverging at these traits and may be experiencing domestication selection. So as an example, we conducted PCA at the 68 loci that are associated with spawn timing. And as you can see, everything's just a big ball. So there's really no separation between the integrated and segregated hatchery lines. And so here in this analysis, we do not observe any genetic change with these trait-associated loci. So no divergence was observed, but that could be due to a variety of factors. So one, Cleveland facility, they maintain best hatchery management practices. So they sample the brood stock across the entire duration of the run. They spawn fish using 3x3 factorial matings. So these best practices that are implemented at the facility may be reducing the potential for genetic change at the different traits. But we can also, another possible explanation is we may have limited power to detect changes for a number of reasons. So one, we're only looking at 9,000 loci, and in our random forest analysis, we only explain approximately 30% of that phenotypic variation. So there's a lot of variation that's unexplained. So we may have limited power to detect changes. And also, we examined 4 hatchery generations, and that's limited time for divergence to occur. So maybe if we continued monitoring, divergence may be occurring in subsequent generations. Lastly, we took our trait-associated loci and we integrated that with the outlier loci that we previously identified. So to remind everyone, we have our different outlier methods that we used, one for the moving average to identify genomic regions that may be divergent, and then also specific loci. And we essentially looked for overlap. So we have a chromosome and an outlier region. If the trait-associated locus fell within that region, then that's certainly suggestive that a trait may be experiencing domestication selection. So overall, out of the trait-associated loci, we observed 6 that were also outliers. And importantly, 4 of these loci were linked to spawn timing. So spawn timing, it was phenotypically divergent significantly in the males between the 2 hatchery lines. And now we're seeing 4 loci that are linked to spawn timing, and they also fall in these outlier regions that we identified. So even though we don't observe any divergence using all 68 trait-associated loci, this is certainly suggestive that spawn timing may be a trait that is affected by domestication selection. We also observed 1 locus that was linked to return timing and 1 locus that was an outlier, and it was associated with both length and weight. And so for the return timing locus, I'll bring back this plot. So here we have a region that's significantly and consistently divergent in the segregated line for all the generations, but it's not consistently divergent in the integrated line. Well, that locus is also linked to return timing. So that, it's just 1 locus, but it's certainly a starting point for future investigations to identify the mechanisms of domestication selection and which traits may be more affected by domestication than others. So in summary, we identified loci associated with 6 traits. We didn't observe any divergence between the 2 hatchery lines when we used all the trait-associated loci, as evidenced by the PCA plot. But we did identify some outliers, some regions of overlap between outlier loci and trait-linked loci. And these are certainly starting points for future efforts. And then really, it demonstrates the utility of genomics. This is an active area of discussion and debate. And in my opinion, I think this study demonstrates the utility of genomics to evaluate conservation strategies. So, does managed gene flow reduce divergence at trait-associated loci that may be adapted? Likely. All right, so overall, just to bring things together, to close, we demonstrated that managed gene flow successfully mitigated genetic and phenotypic change associated with captive breeding across 4 generations. And these studies can further inform best hatchery management practices throughout the region for risk scenarios, and it also provides, because we compare 2 contrasting regimes over multiple generations, it provides insight into the rate of divergence, among other things. And we also characterized, using these novel genomic approaches, we characterize multiple genetic risks, including some of the first regions that may be under domestication selection in Pacific salmon. We linked the risks to fitness-related traits. And really, there's a lot, this research, a lot more questions that this research brings to mind, and so it really lays the foundation for future efforts. All right, so that's it. Just got the knowledge and select. Conducting good research requires a healthy mind and body. With that, I'd like to thank This was really a big collaboration between a lot of people, and so I'd just like to acknowledge my co-authors. So my co-authors, Maureen, Jeff, Dave, Bill, and Kurt. So Dave, Bill, and Kurt were phenomenal. Like, I was pestering them so many times, asking for data, asking for clarification about the system. Yeah, really everybody, they contributed significantly, and this work would not have been possible without them or anyone else that I'm about to acknowledge. Data collection, lab, and analytical assistance. A lot of groups of people, folks from the Yakima Nation and WDSW, of course, they were the ones that really collected all the data, and we were allowed to subsample the data and use it for these studies. A lot of folks, Garrett, Karita, Tara, Jim Thorson, Mackenzie, Carolyn, Krista, and the entirety of MERLab, my lab group, they all provided integral assistance in both data analysis and interpretation. And I'm realizing this now. I left probably the most important person off the list, Isadora, our lab manager. She taught me everything that I know in the lab, which isn't much. I still have a long ways to go. But she was absolutely integral. For the first 2 weeks of grad school, I was pipetting incorrectly. So she corrected me there. Then also funded all these funding organizations, NOAA, Washington Sea Grant, big thanks to them. The NOAA Sea Grant Population Dynamics Fellowship, and as Kerry mentioned, the University of Washington Hall Conservation Genetics Research Award. PhD committee. Kerry, you've been phenomenal. Hold on something. Yeah, you've been a great mentor, fantastic scientist. You've really, I don't know, words cannot describe how much you've impacted me in my science. That's like my development as a scientist, and so I really appreciate it. Jeff, this is old school Jeff photo from Philipsport Waltz back in the day. Jeff has become, our de facto modeling expert. He denies it. But Jeff has helped us in all these studies to a great extent. Steve Schroeder, you've provided excellent insight into both the background of Cleellum and the biology of the system. And I wish I had used you. I wish I had tapped into your knowledge more. But hopefully we can continue to work on some of these issues in the future. Brian Kelly, my new GSR, last minute. Yeah. Because Tim Thornton was my old GSR, so graduate student representative. But I didn't have funding for this quarter, and Tim, because some of the areas of human genetics overlap, like in the skills overlap with some of the stuff that I'm doing. He took me on as an RA to work on a really cool project with Alzheimer's disease in humans. So he could not be my GSR once he was funding me, but it's been a pleasure. It's been phenomenal. Yeah, Maureen Purcell. The disease chapter did not make it in, but I had a great time working with you at USGS. Y'all are hiring? Like those analyses, they're in the works, so we'll be working on it really soon, and I'll be pestering you, I'm sure, soon enough. And also Lawrence Hauser, so he's trying to fit into his old wetsuit here. You've been great. Right now, you've been a great resource. For their genetic theory and interpretations. And so, yeah, I can't thank my committee enough. And they've also been very flexible with me and my schedule. MERLab, MERLab, sort of the old school edition with Shannon, Miyako, Jocelyn, Dan. Yeah, they were, integral in when I first started. Maureen and Dan in particular, I was bugging them multiple times per day, but they were always helping me with a great attitude. If Maureen's watching, thank you very much, Maureen, all the way from Norway. Then the current MERLab, Mary, Sam, Natalie, Molly, and Lenny, and Isadora. Yeah, you've been great. I do not want to accept any job unless everybody can come with me. Both like emotional support, just friendship, it's been great. And lastly, my family and friends. So I'll start off by acknowledging Jake in the back, my running buddy, my maker of spandex running shorts, unofficial life coach. So thank you for all your friendship and support over these years. My family, my parents are here. They certainly, I wouldn't be who I am today without them. And then Emma, my girlfriend Emma, like amazing. So thank you for that. So that'll take any questions.
penetrating question. Nice.
So now that there's genomes available for Chinook, are you going to go back and look at any of these outliers and see if they're near these particular interests?
So that's a great question. So the Chinook salmon genome came out earlier this year, so it enables us to align a lot more of our loci to the genome and get a better idea of that. I would like to, if there's if anyone wants to pay me to do that. So I would really like to. I think that would be a great project for another grad student or for myself. But I would have to obtain funding from somewhere. But yeah, definitely.
So the
our congratulations to Charlie. As you said, Kerry, it's been wonderful to work with the guy. He's been very cooperative, very collaborative, come over whenever we wanted to hear results or present those results to us. So it's just been wonderful working with him. Anyway, just wanted to say that. And if anybody's interested in working on the follow-up questions that are ready, we would be happy to work with you as well. Good job.
Thank you very much. We are the interlopers on the system. Thank you. Yeah. I'm guessing that I know Ryan had a question. Ryan's on the committee.
It was more of a general interest question.
So let me just tell you is that right now I'm going to ask Charlie's committee to stay here to discuss his thesis with him. Charlie actually said that he didn't want to do anything major after his thesis because he thinks he's got lots of corrections and he's going to hand in his thesis tomorrow. So if you guys would like to commiserate with Charlie afterwards, we should be back in about half an hour to an hour in our lab section, and we've got things that we shouldn't have.
Thanks all for